Today we are going to talk about the foot and ankle of Australopithecus sediba, a research paper by Berger and colleagues. This presentation was created by Melissa Rudo, Dr. Ibrahim, Jack Gibson, and Andrew Grada.
Aside from our large brains and capacity for intelligence, one of the most defining characteristics we share as humans today is our upright posture and bipedal mode of locomotion. Because of the growing number of foot and leg elements being added to the fossil record, researchers are beginning to notice more and more morphological diversity between the specimens from extinct and modern hominids. This information prompted Berger and others to attempt to gain more insight on characteristics of foot configurations that allow for bipedal locomotion. This was achieved through analysis of well-preserved and well-articulated foot and ankle bones of the extinct hominid Australopithecus sediba. The taxon Hominidae is a family of great apes including chimps, orangutans, gorillas, and humans. The Australopithecus sediba lived about 1.98 million years ago, and based on where fossils were found, they resided in South Africa around the infamous Malapa fossil site. Berger and his colleagues used specimens from this extinct species in order to discern what they imply about their locomotion, as well as whether their morphologies were more primitive and ape-like or more convergent to the characteristics of derived Homo and modern humans. The specimens studied included the distal tibia, talus, and calcaneus, cemented together with calcified sediment, as well as the right distal tibia of another organism, the right fourth metatarsal, and the right proximal fifth metatarsal.
Because the specimen is cemented in a matrix, any attempt to physically detach and reassemble the specimen would result in severe damage to the morphology. So the researchers went around this by utilizing medical computed tomography, which allows the researchers to digitally splice up and separate the matrix in order to make a 3D rendering and compare those bones to apes and modern humans. Here is a virtual reconstruction of the previous matrix, right distal tibia, talus, and calcaneus.
By comparing the virtual 3D images of the foot and ankle bone of Australopithecus sediba, two main insights become apparent. The first main insight is that Australopithecus sediba shows signs of both ape-like characteristics and modern human bipedal motion. In the graph shown here, Australopithecus sediba is represented by red dots, modern humans are represented by white squares, and shaded diamonds represent members of the genus Pan. Function 1 represents movement due to the part of the ankle by the anterior width of the tibial plafond and the thickness of the medial malleolus, which are found on the top and side of the ankle joint respectively. Those with greater function 1 are graphed on the right. As you can see, Australopithecus sediba have low function 1, categorizing them with modern humans more so than members of the genus Pan. In addition to the similarities in function, the overall shapes of the feet are arched in a similar way to modern humans. Further analysis shows some differences among Australopithecus sediba and modern humans. The overall forms of the ankle and foot bones suggest that Australopithecus sediba stood upright. However, the neck angle and head torsion angle are more reminiscent of related hominids, such as Australopithecus afarensis, as shown in the picture. The second major insight gained from this study is that Australopithecus sediba may have contained a degree of arboreality, or living in and among the trees, due to their similarities to primates. In the diagram, the peroneal trochlea is indicated with a dashed oval, and the black arrows are pointing to where it is connected to the lateral plantar process, or LPP, which enables primates to increase the surface area of the heel to aid in force dissipation. As you can see, modern humans have an orientation where the LPP is on the bottom of the heel, and the MPP, or medial plantar process, is not visible at all. With this structure already present among juvenile Homo sapiens, which can be seen on the bottom right of the diagram, Australopithecus sediba have much more ape-like structure, with both the LPP and MPP being present, which may allow for easier movement among the trees. As shown in the bottom left of the diagram, these are already present among juvenile Australopithecus sediba. This diagram plots the data collected by comparing the differences in LPP and MPP structure, and this shows that Australopithecus sediba is more similar to primates than humans in that regard. It is because of this info that there is a chance they contained arboreality similar to their ape ancestors.
The concluding thoughts that we gathered from this research paper is that the evolution of human bipedalism is likely to be more complex than we previously thought. For example, the differences between the Australopithecus sediba foot and the Australopithecus afarensis foot support the idea that the acquisition of bipedal adaptations in the hominin lineage evolved in an intricate fashion and is more complex than we assumed. This means homoplasy, which is a character shared by a set of species but not present in their common ancestor, could be implied for human bipedalism based on the combination of features in Australopithecus sediba. From this case study, the researchers give two possibilities that can be inferred. An ancestor-descendant relationship between the Australopithecus sediba and a direct or transitional species such as the Australopithecus africanus is unlikely, indicating a more distant common ancestor. The second possibility is that if an ancestor-descendant relationship did exist, it would require evolutionary reversals in calcaneal and distal tibial morphology, despite habitual bipedal locomotion in both species. Lastly, some final thoughts we concluded is that the more fossils that we find, the more complex the evolutionary picture looks. The researchers could have expanded the study by analyzing leg bones as well as ankle and feet bones, or conducting a more exclusive study comparing Australopithecus sediba and Australopithecus afarensis. One last question to think about is how will future discoveries further impact our understanding of the evolution of modern human bipedalism?